All right, let's get rolling. So welcome to our webinar on enriching engineered T cells with antibody sequencing and modulation about next generation proteomics. I'm your host, Ian Rogers, and I'll start by going over our agenda today. We'll be talking about enrichment strategy for T cells, applications of protein sequencing, and how some proteomics techniques work in the cell therapy context. I'll start by introducing our speakers today. Patricia Hernandez-Lopez is a PhD candidate with Professor Jürgen Kabel's group at the University Medical Center in Utrecht. She holds a master's degree in therapeutic targets and cell signaling R&D and has industry experience in molecular biology and cancer research. So thanks for joining us today, Patricia. Monique, my colleague, is a scientific sales executive with Rapid Novor. She holds a master's of science degree in biotechnology and management from the University of Glasgow. She has years of experience working in proteomics industry and was actually a featured speaker recently at the Pivotal Links conference in Oxford. But let's start with some context before we jump into Patricia's presentation. By the numbers shown here, we can see that cell therapies, and in particular CAR-T and CAR-NK, are becoming increasingly important, especially in the fight against various cancers. The premise is to extract a patient's own immune cells, of course, engineer them, produce them in larger quantities, and then reintroduce them back into the patient. We can think of this as essentially basically an individual manufacturing run every time. But the challenge is it's with variable inputs and a requirement for high quality, safe, effective outputs. So if you look at that as the overall challenge for the industry is to manage production costs while optimizing process controls. So the more extensive the process, the less economically viable is the treatment for the patient or for the company itself. So the most promising angles here are looking at designing better chimeric antigen receptors, or CARs, automation in manufacturing, and innovation in process controls. So let's look at that. When it comes to designing better CAR, the optimal CAR structure for each given antigen to be targeted involves the generation and testing of several constructs, and some would say an unreasonable number of constructs, given the complexities of the different types of CAR designs. When it comes to optimizing production process for cost efficiency, efficacy, safety, one strategy that's gaining attention is enrichment of the product. And we're going to talk about that today. So one can create process reagents that bind to engineered versus natural cells. In both cases, understanding protein sequences and structure can help, and in particular, rational design based on antibodies known to function against the target or the T cell receptor itself. So why don't I now turn things over to our client to take us through just such an enrichment strategy that they developed.
Hello, everyone. My name is Patricia, and I'm a PhD candidate in the group of Jürgen Kubal and Sol Sebastian at the University Medical Center of Utrecht in the Netherlands. Today, I would like to show you this paper that we published end of last year about the characterization and modulation of anti-aßTCR antibodies and their respective binding sites at the beta chain to enrich engineered T cells. So adoptive transfer of TCR gene-modified T cells has been proposed as an attractive approach to target tumors. However, only a small proportion of the patients can actually benefit from this kind of treatments. So one of the reasons that has been described to explain this is the presence of non-engineered or poorly engineered T cells in the final product that is administered into the patients. So the presence of these non-engineered T cells in the product will not only impact the clinical outcomes, but also could lead to toxicity. So some reasons for this are the insufficient expression of the introduced receptor or mispairing of the introduced alpha-beta TCR with endogenous alpha-beta TCR. So actually, as you can see, giving the patient a really pure product will not only help with the effectiveness of the therapy itself, but also will help the therapy to be safer. So what we did actually in this paper was just to develop a method for the purification of engineered T cells to increase efficacy and also safety of the alpha-beta TCR gene therapies. So actually what we did was to just develop a strategy in which we can enrich engineered T cells after being transduced with an alpha-beta TCR. In this case, we use an anti-tumor alpha-beta TCR. So what you get when you transduce T cells that, for example, come from a patient, you will get some cells that are non-engineered at all, some cells that are poorly engineered, and some cells that are nicely engineered. So ideally in your product, you would like to have the third type, so the cells that are really nicely engineered. So to develop this strategy, we actually base it on the use of this GMP grade anti-human alpha-beta TCR antibody. And the idea behind this is that we will introduce some mutations in the TCRs that we are using to transduce these cells in a way that this antibody will not be able to bind to the TCR anymore. So this means that when we use this antibody, it will bind to the cells that are not transduced or poorly transduced and express the endogenous TCR. However, it will not be able to bind anymore to the cells that are nicely transduced and express the introduced TCR. So then you can think of using, for example, max sorting to deplete the cells that are poorly engineered or non-engineered at all. So that you will end up with a product that is actually very pure for the cells that have been engineered. So for that, the first thing we did was to just study where this antibody was binding in the TCR. And for that, yeah, so you can think that if this is an anti-pan alpha-beta TCR antibody, you can think that it will bind to the constant part. So the first thing we did was to actually test this. And for that, we create a chimeric TCR in which the variable part of the TCR is human, while the constant part of the TCR comes from mice. So then we transduce this into Jurkat cells. And what you can see is that when you use an anti-human alpha-beta TCR antibody, and you have the fully TCR coming from mouse, this antibody is not able to bind. When you have the fully human TCR, the antibody is able to bind. However, when you have your chimeric TCR in which the variable part comes from human, but the constant part comes from a mouse, this antibody is not able to bind anymore. So this will confirm that this antibody is actually binding to the constant part of the TCR. The next question is, okay, which chain is this TCR binding? So is the alpha chain or the beta chain? So for that, what we did was to just study the sequences of the human and murine TCRs for the alpha chain and the beta chain. And what we observed is that there were some parts that were non-homologous between both species. For example, in the case of the alpha chain, there are 3 regions that are non-homologous, while in the case of the beta chain, there are 4 regions. So what we did was to just substitute these different regions for the mouse counterpart in the human TCRs, and then we create different variants. So for example, in the alpha chain, 3 variants, and in the beta chain, 4 variants. Then we transduce this into Jurkats again. And what you can actually see is that when you modify the alpha chain, you actually don't get any change in the binding of the antibody if you compare with the fully human TCR. So you see that the binding is the same for all the variants. However, when you modify the beta chain, you observe that when you modify this N3 region here, that it was located actually here, what you can see is that modifying this region, you actually completely abrogate the binding of the antibody if you compare to the wild type TCR. So then we know that the antibody is binding to this region, but you wouldn't like to give a patient a TCR that contains so many mutated amino acids into mouse, because this could be really immunogenic for the patient. So what we did next was to try to reduce the number of amino acids that we mutate to the maximum as we could. So then the next thing we did was to actually look into this M3 region, and again we compare human to murine sequences, and we observed that there were 11 amino acids that were non-homologous between both species. So then we created mutants with all these amino acids, and what we observed is that 3 of them could actually reduce the binding of the antibody. However, none of them was able to actually completely abrogate the binding, as it happened when you have your full M3 region mutated. So then we thought that maybe combining these 3 different mutations, we could abrogate the binding of the antibody. So we did that, and we transduced again T cells, and what you can see is that indeed when you mutate when you introduce this D112G mutation together with one of the other ones in the TCR, the binding of the antibody is completely abrogated. So then for the next experiments, we just select this mutation just as a proof of principle. And the next thing we did was to actually check if the strategy that we have developed was actually working. So for that, we just transduce like a wild type TCR and a TCR that was including the 2 mutations. So what we did after transduction, we used this strategy using this anti-alpha beta human TCR antibody to deplete the cells that were not transduced. And what you can see is that when you have your cells before depletion, you get like around 28% of your cells are transduced, as is shown by the binding of this B beta 4 antibody. However, after depletion, this percentage increased to almost 88% of the cells transduced. So you can see that this method is actually working quite good, and this can also be shown by the binding of the NY-ISO1 pentamer to the TCR. So then you can really see that this depletion significantly increased the percentage of positive cells for the pentamer staining in the product. So once we know this strategy works, we try to combine it with other strategies that has been described by others to increase the TCR pairing, and just reduce the mispairing. So we selected these 3 strategies. So the first one is actually described by Sommermeyer in 2010. And it actually, what he did was to just introduce some murine mutations and create minimally murinized TCRs. So in this way, you increase preferential pairing of your TCR. Other option that you can do is like to introduce an additional cysteine that will increase the pairing, or also introduce this additional cysteine together with the transmembrane domain of alpha gamma delta TCR. So what we did was to actually use these 3 strategies and add to these ones our 2 mutations. So then we transduce our T cells again with these 3 TCRs together with that, all of them include our mutations. And what you can see is that while that actually there are not super huge differences in mispairing reduction, however, you can see as by the ratio of B beta 4 and double positive cells, so alpha beta TCR plus B beta 4, that the one that seems to reduce as like more the mispairing is the combination of a cysteine together with a gamma delta TCR transmembrane domain. So the next thing we did was to try our strategy with all these cells, and you can see that in all the cases we got increased percentage of B beta 4 positive cells, so transduced cells, after depletion. So it significantly increased the percentage of transduced cells in the product. So the next thing we did was to just use these cells and test them functionality to check if actually if this depletion helped for the function of these cells. So for that we check interferon gamma release, and what you can see is that in all the cases the depleted cells show like increased interferon gamma release compared to the non-depleted ones. So this not only happens when you use a NY-ESO peptide to pulse your cells, but also with cells that actually express this peptide by themselves, just like endogenous expression. So the last thing we did in the paper was to actually try to use these mutations that we introduced in the TCR to not deplete like the non-engineered cells, but to actually deplete the engineered cells, because you can think that when you give this kind of therapies to a patient, sometimes something can go wrong. And for example, you can have like cytokine release syndrome. And then ideally you would like to have a way to remove these cells from the patient. So then we thought that maybe using an anti-mouse TCR antibody, so that just bind mouse TCRs, we could deplete these cells. So that's what we tried. And then what we did was to test the binding of an anti-murine TCR, beta TCR antibody against these cells that we transduce. And what you can see is that actually this antibody, this mouse antibody, is not able to bind to the TCR. So then we came back to our comparison between human and murine. And if you remember, there were 11 amino acids that were actually non-homologous between both species. So what we thought is that maybe increasing the number of antibodies, we could use this strategy not only to enrich for engineered cells, but also to deplete them in vivo from patients. So we created 2 new variants, one in which we mutated 9 out of 11 amino acids, and the other one in which we mutated 11 out of 11 amino acids. Then we test again the binding of the antibody, and what you can see is that for the 9 out of 11, it actually binds quite good. Surprisingly, we found less binding for the 11 out of 11, and then it seems that the introduction of these mutations affects a bit like the electrostatic forces. So that seems to be the reason for this lower binding. So the next thing we did was to try to humanize this antibody, because it's actually a hamster antibody, so it's producing hamsters. So that's again not nice to administer into a patient. So that's what we did. And then what you can see is again that this chimeric antibody, you can see that it binds equally nice than to the completely to the murine antibody or hamster antibody. So the next thing we did was to just conjugate this antibody to a drug that is called MMAE. And we just treat these cells with increased concentrations of this antibody bound to the drug. And then what you can see is that 9 out of 11 variant, you can see that you can actually deplete your cells at the highest concentration. So we know this is far from being perfect, but it's more like a proof of principle. And of course, this would need probably more engineering to get better results. But it's actually quite nice that you can use these mutations to get both, like to engineer your to enrich your product, but also to deplete the cells from patients when something goes wrong. So just to conclude, here we describe a novel strategy for the enrichment of primary alpha beta T cells engineered with a murinized alpha beta TCRs using a GMP-ready anti-alpha beta TCR antibody for the depletion of non and poorly engineered T cells. Also, we show that additional murinization allows for depletion of alpha beta T cells engineered with a murinized alpha beta TCRs using a chimeric anti-mouse TCR beta antibody. So I would like to thank to all the authors that helped to get this paper published. And also here, if you want to read the full paper, you can just access via this QR code here. So thanks everyone for listening.
All right, thank you so much, Patricia. It's really great to learn about some of the work yourself and your team have been doing. we're really happy to have you as clients and, definitely looking forward to working with you in the future. So before I explain how our services can be used in the context of cell therapies, I'll just provide a bit of background on who we are. So we are a Canadian-based proteomics company, and our ultimate goal is to decode immunity. Now, with this mission, we offer a variety of services in the proteomics space, many of which are based on next generation protein sequencing. I'll explain this technology in a little bit, but it is very different from admin or N-terminal sequencing, which is used more for protein identification. Now, through our applications of next generation protein sequencing, or NGPS, we've developed the largest privately owned MS-based proteomics company in Canada, and we've sequenced over 5,500 antibodies and proteins. We're also really proud to say that we're the first team in the world to sequence antibodies from Thera using only proteomics. Now, before I get into the actual tech, I've thrown our service offerings on the slide for you there. The first three that you see surround sequencing work with antibodies, but we can also do it with non-antibody proteins. So we started off with monoclonal sequencing and have since developed polyclonal sequencing, where we can actually pull out heavy and light chain mAb pairs. And we have MatchMab, which is our sequencing verification service. And we can also offer some extra bit of analysis there about protein variations or post-translational modifications. We started offering epitope mapping just last year as we were getting some requests for it, and it fits quite nicely into our repertoire of protein analysis. And last but not least, we have a proteomics software for those that have some MS data on peptides as well. So our proteomics services can get quite technical, and to avoid putting you all to sleep, I'll do a high-level overview. At its core, our tech involves the use of mass spec, bioinformatics, and proprietary machine learning algorithms. Now, for our monoclonal sequencing service, REMAB, we take purified protein, digest it using a variety of enzymes, or our protease cocktail, as our team likes to call it, and we throw the array of peptides through one of our many mass specs. This data is then analyzed by our algorithms, and at least 2 of our bioinformatics specialists take a look at it as well. When they're satisfied that our data quality standards have been met, the data is put together in a tidy report detailing variable regions, CDRs, and some DTMs as well. Now, what's so special about Rapid Nova's NGPS? Well, we have industry-leading quality standards. So with 30-fold coverage over every single amino acid within your sequence, this results in a 99.9% confidence. And we also have the ability to differentiate between isobaric amino acids, isoleucine and leucine. Typically, these 2 amino acids can be difficult to differentiate because they have the same mass. However, we have what we call our WILD technology, which stands for W-ion isoleucine to leucine differentiation. So basically, we can break off the side chains from the amino acids, and the ions have different weights. And this allows us to differentiate between the 2. Now, this technology is important, especially when looking at mutating specific amino acids or to change binding. All of our work is done in real time, so we can adjust MS settings as we're getting the data to optimize the accuracy. And the resulting data from our proprietary algorithm is also reviewed by at least 2 of our bioinformaticians to ensure that every single amino acid has the appropriate coverage and meets our quality standards. And finally, what's unique about our technology is that, whether it's a hamster, a mouse, or a human, or if it's an antibody, IgM, IgG, whatever you throw at us, with our track record, we've likely seen it before, and we can definitely sequence it. Now, our requirements for REMAB are quite simple. We ask for a small amount of sample with at least 80% purity. The purity is important as when we're working with mass specs, we want to reduce any background noise, as contaminants can negatively impact the quality of the mass spec reads, and as a result affect the quality of data that we have to prove the sequence is in fact the sequence. Now, with our polyclonal sequencing technology, it's a bit more complicated than our monoclonal sequencing, as we're pulling out paired chains from polyclonal mixture. So there are 2 ways that we can accomplish this. The first is through a proteomics technique, which is the branch on the top there. And we use our mass spec algorithms and specialized team. And we also have a proteogenomic approach as well, where we actually combine both branches that you see there. So we include NGS data, and we're able to pull out sequences in that method as well. So we don't do the immunizations or the production of recombinant antibodies in-house. We work with partners for this, or sometimes our clients have in-house capabilities. But the monoclonal antibody chains that we pull out from the polyclonal sera are generally the more dominant forms in the polysera, and based on what we've seen, they have really strong affinity binding. They're actually binding better than, if not the same as, the original polyclonal mixture. So they could be great for obtaining Fabs for the antigen binding domain as well. In regards to our epitope mapping, we use HDX MS, which measures the exposure of hydrogen molecules on the surface of a protein or protein complex. So the first step causes hydrogen molecules to be readily exchanged with deuterium. Hydrogen molecules that are buried within the structure, for instance, at the binding site of an antibody or antigen complex, they're generally not exchanged as readily. So when we examine the intact proteins and proteins via mass spec, and then we compare them with the amino acid sequence, we can then determine which regions are the possible binding locations. The first step is labeling. So you'd incubate different fractions with DTO solution in a series, in a time series, and then through quenching, you'd control the temperature to prevent further exchange or back exchange. The next step would be to digest and prepare peptides for analysis. So this would be run through the LCMS, and then finally the results would be analyzed, and the deuterium uptake of each amino acid, as well as the binding sites, would be identified. This can be done with similar samples. So for example, antibodies with similar sequences. Now, I've given you a brief overview of some of our services, so I'll just lay out how they could be integrated into the cell therapy workflow. So for research and target ID, REMAB can be used here to begin research on known binders, or REPAB can be used to assess a native immune response to a target. In regards to MAB discovery, REPAB can definitely be used as a method of antibody discovery for novel antigen binding scFvs. We can examine the functional native immune response of both patients or animals here. And REPAB can also be used to secure existing known binders as well. For the TCR CAR engineering, having the antibody sequences of nanobodies or Fabs through REMAB helps to direct specific engineering to improve affinity or specificity of the antigen binding domain. And additionally, through our HDX Epitope Mapping, we can provide insight on linear, structural, and conformational epitopes, which can help binding site characterization for engineering. Finally, in regards to the production process, REMAB can be used as a part of antibody-based enrichment. So by sequencing an antibody, site-specific mutations can be created once again in order to enhance selectivity and binding affinity, as Patricia demonstrated. So overall, having the protein sequence aids in TCR design, and the sequence information can also contribute to the improvement of the quality of enriched cells before infusions. Higher quality enriched cells, as mentioned by Patricia, can lead to more effective treatment and ideally better clinical outcomes for patients. And by having direct access to the protein and applying NGPS, you can have greater assurance of the sequence of the molecule that you're working with, including differentiating isoleucine and leucine, which is important when changing specific amino acids for targeting enriched molecules. And finally, it is useful for epitope and protein interactions, can help provide binding insight and help guide enrichment strategies. So that brings me to the end of my presentation. Thank you all for tuning in, and I'll now open up the floor for some questions.
Thanks so much, Monica, for your great presentation there. Yeah, please use the Q&A function at the bottom or the top of your screen, depending on how you have your Zoom set up, and to enter any questions in. and we'll do our best to address them. I don't see any questions on the chat just yet, so we'll give everyone a minute to see if there are any questions that evolve. Otherwise, we can wrap up here and certainly address any questions that you might have by email afterwards. Well, not a question, but a comment from Alexandra. Great technology. Thank you so much. I appreciate that. Okay, but yeah, we're very happy to see mass spectrometry used like this as well. There's other new and exciting things coming out of our labs too, which we're excited to share, and can't wait to see them on our services list coming up here. So stay tuned.